Hi everyone, today we're going to begin Unit 4. We're starting with the first lecture, which is Introduction to the Animal Kingdom, or Chapter 32, which is Animal Diversity. So we're going to be starting off on page 85 in your lecture course pack, and this is the summary of what we're going to be learning in Unit 4. Today's lecture will focus on identifying key characteristics common to all organisms, and we're going to learn some background information on animal evolution and traits that are common amongst various animal phyla, specifically different body plans. So you will be taking notes in your lecture course pack starting on page 86 through page 90. And so we're going to begin with features that all animals have in common. All right, so when we look at these animals right here, right, we've got Kesha, we've got little kitty, fish, etc. And we want to think about things that they all have in common. Now, the cat and Kesha, they are vertebrates, so they have backbones. And these animals over here, our fish is a vertebrate too. A vertebrate means you have a backbone, or vertebrae. And our invertebrates don't have a backbone. We're going to be learning about invertebrate animal phyla in our next lecture, and then our third lecture in this unit will be on vertebrates. And so animals don't have a backbone in common. That's something that will only be common to only vertebrates. So what do animals have in common? And so here are a list of features that are common to all animals according to your book. So your book says animals are multicellular, heterotrophic, eukaryotic, and they have tissues that develop from germ layers or embryonic layers. So I want to break that up into some different categories and expand on some of these a little bit more. And I also want to compare them to other eukaryotic lineages like protozoa, fungi, and plants. So we're going to compare animal traits to other types of eukaryotes. All right, so nutritional mode. Heterotrophic means they get their organic matter from food. They don't make it like an autotroph would. Heterotrophs. Okay, so animals are heterotrophs, but so are protozoa and fungi. Plants, of course, are autotrophic. So what makes animals different from some of these organisms is that animals ingest their food. Fungi don't ingest their food, they absorb it. Protozoa, if you count phagocytosis as ingestion, they will ingest their food. But the protozoa and animals are going to be different in other ways. So for cell structure, they are eukaryotic and they are multicellular. So at the cellular level, they are eukaryotic, meaning the cells have a nucleus and organelles. All of these are eukaryotic organisms as well. But in terms of cell structure, the organisms are multicellular, meaning they're made up of more than one cell. Protozoans are single-celled organisms, so this is one way now that protozoa are different from animals. Fungi can be single or multicellular, and plants are also multicellular. All right, and cells are specialized into tissues. So tissues are going to be groups of specialized cells. We're going to go over what tissues are in more detail in development. But for right now, we'll just say that protozoa and fungi don't have tissues. Fungi will have specialized cells, but technically not tissues, whereas plants do have tissues. So reproduction is, animals are characterized as undergoing sexual reproduction. There are some animal species and animal phyla that can reproduce asexually, but as a whole, all animals can reproduce by sexual reproduction. And so just to review what sexual reproduction is, it's when you have two gametes, haploid cells, that fuse together in a process called fertilization, and they form a diploid cell. So that's sexual reproduction, and we have that as a common feature amongst all of these eukaryotic organisms as well. All right, so all animals can reproduce sexually. Some will be able to reproduce asexually, and we'll note that when it happens. And in terms of development, we're going to right now stick with the fact that animals have tissues, which are those specialized cells separated by membranous layers, that develop during early embryonic development from something called germ layers. Embryonic development. And so this is not a feature found in protozoa or fungi, but only in plants and animals. All right, so this is some characteristics that all animals have in common. Throughout the next several lectures, we're going to be talking about things that make different animal phyla different. But these are things that all animals will have in common. Okay. All right, so let's go over that early embryonic development. Early embryonic development just refers to the stages of a zygote becoming an adult animal. So we're going to start with the zygote, which is simply a fertilized egg. All right, so the fertilized egg comes from two gamete cells that have fused together. So fertilization would be the process of forming the zygote. Oh, here's that definition right there. All right, so the first stage, or for the first process, so our processes are going to be found in italics. Our first process is called cleavage. And cleavage is a series of mitotic cell divisions that will occur after fertilization. So it'll be the process that occurs between the zygote stage of the embryo and the blastula stage. And so cleavage reactions form, one cell will form two cells will form four cells will form 8, etc., until you get about 64 cells or even more. And then we have a hollow ball of cells called a blastula. So a blastula, I'll often just call it a hollow ball of cells formed from cleavage reactions. During embryonic development. All right, and then this center space right here, this fluid-filled center of the blastula, that is called the blastocele. And every time we see that suffix or that word cele, this is going to refer to a cavity or a space of some sort. All right, so you have a room in your course pack to draw this. So you're going to take a moment to draw what we just did, and we are going to focus on the terms at the bottom of page 86 and at the top of page 87. And so we're starting with our zygote, which is the first stage of embryonic development. That is our fertilized egg. It goes through a series of mitotic developments to form a blastula. And so I'm just going to draw my cells in a two-dimensional version around the edge here. All right, so after mitosis, this process is called cleavage. We now have our blastula, and that space in the center is called the blastocele. All right, so that is the first stage or the first process of embryonic development. And one other thing I want to note is you're going to see the word embryo a lot. So an embryo refers to any stage in the early processes of embryonic development. So a zygote is an embryo, a blastula is an embryo. These are just different stages of embryos before and after cleavage. All right, the second stage is going to be gastrulation. So gastrulation will be what happens next. Here's a picture showing that process of gastrulation. So we're starting with the blastula, and then what we're going to see is a folding in of one end of that blastula. This picture is nice because it shows the three-dimensional version of it, whereas my drawings will be in 2D. So in this folding in process, we're now going to have two spaces. We're going to have the inside space, which is called the blastocele, right? So if this is the blastocele right here, that's going to become the blastocele right here. But now we have this space right here, and this space, the inside, or the inside that's exposed to the outside, that's going to be called the archenteron. So we now have two types of cavities, the inside cavity called the blastocele, and the archenteron cavity, which is the cavity that is exposed to the outside. All right, so we're going to draw this again. So we are starting now with our blastula. One end is going to get folded in during the process of gastrulation. And we are going to have, I kind of like to draw it, and I consider it sort of like an airplane neck pillow, right? It fits right around your neck for a comfortable airplane ride. All right, so this, I'm going to draw my cells now. We're going to have different layers of cells in our gastrula. This outer layer of cells is going to be called the ectoderm cells. Ecto means outside, so these are the cells of the gastrula that are exposed to the outside. I'm going to switch colors. Pink. All right, so now we have the cells that are facing the archenteron. These cells right here are going to be the endoderm cells. So endoderm and ectoderm are our first two germ layers. And the endoderm is going to become the tissues that are the lining of the gastrointestinal tract. So our stomach and our intestines, the lining of the stomach and intestines come from our endoderm germ layer. The ectoderm germ layer, this is going to become our outer cells, or our outer tissues, like our skin, and also our nervous system. So our tissues that we have in our bodies derive from these two germ layers right here. So ectoderm and endoderm are going to be our germ layers that are formed during gastrulation or from gastrulation. All right, and so this space in here is the blastocele, and that becomes this space in here. So we have two blastoceles, one in the blastula and one in the gastrula. And so now the blastocele in the gastrula is going to be in between the endoderm and the ectoderm germ layers. And then this space right here, this cavity, this is going to be called the archenteron. And this is going to later become the gastrointestinal tract, or the gut. And so this opening right here, that opening is called the blastopore. And the blastopore becomes the opening to the GI tract, and eventually it will become either the mouth or the anus. All right. So I think that covers all of our terms in cleavage and gastrulation, the first two processes of early embryonic development. Here's a video that shows that happening in a sea urchin, which is an echinoderm. Hold on. So you're seeing some of those cleavage reactions as the cells start to divide from the zygote stage. Early cleavage reactions, and now we've got our blastula, blastocele on the inside. We're going to see some new cells starting to form at this end, and we're going to see that folding in process during the first stages of gastrulation. There's that folding in, and we see the archenteron forming. And it'll continue to elongate as it expands throughout the gastrula. Right. So here's a two-dimensional version of what we just saw. The zygote becomes the blastula during the cleavage stage. The blastula becomes the gastrula during the gastrulation stage. In some animals, we are going to see the archenteron, or these endoderm cells, go all the way through to the other side. And so in animals where this happens, we're going to have a complete digestive tract that will go from mouth to anus. And in many of these animals as well, we're going to have a third tissue layer called a mesoderm tissue layer, which we'll get to later on in this lecture. All right. So now we're going to move on. We're at the bottom of page 87 in our course packs. So at the bottom of page 87, it asks you to review the important order of events in the history of life. And so we're going to start with, obviously, the earth formed, but then in terms of life, we've got the first prokaryotes that appear on the planet. So prokaryotes evolved first. The second important thing that happened is that oxygen started accumulating in the atmosphere. And so that happened much later after the evolution of the prokaryotes because of the cyanobacteria making oxygen through photosynthesis. Our third important event was the evolution and appearance of the single-celled eukaryotic organisms through the endosymbiotic theory that we learned about in Unit 1. After a great period of time, multicellular eukaryotes like fungi and certain types of algae started forming. And eventually then we got the first animals. And so this is going to be between 700 and 800 million years ago. Yay! So this is where our lecture begins, the formation of animals, or our unit, I should say. And then somewhere before land colonization, but in the very early stages of the Paleozoic era, during the Cambrian period, we had the Cambrian explosion. So a great expansion in animal diversity. So it's not on this chart here, but this is an important event in our timeline that happened before the colonization of land. And so land colonization happened first for the invertebrates, inverts made their way to land first, and then the vertebrates. All right, and so that would be our last important event in our timeline. All right, so now we're going to turn the page to the top of page 88 in our course pack and fill out some information about those first animals. So our first animals were the sponges, or members of the phylum Porifera. They evolved about 700 million years ago, but it's really more like 700 to 800 million years ago are the estimates. Some of the charts you'll see later will say 770 million years ago. And the common ancestor that was shared with the first of the animals were the choanoflagellates. So the choanoflagellates had a common ancestor with sponges and other early diverging animal lineages. So choanoflagellates were a group of single-celled protozoa, and they were colonial, which means they would kind of join together and form a colony. So the individuals would sort of aggregate together. And these colonial choanoflagellates, which were protozoa, are very similar in structure to a cell that's found in sponges called the choanocytes, or the collar cells. So they're very similar in their structure. And so what happened for the ancestor, so the ancestor would be right here, this ancestor diverged in one direction to be the choanoflagellates, and something else happened to become or evolve into animals. All right, so two mechanisms had to evolve for that jump between the common ancestor and our animals. And those two things is that the animals had to attach to one another, not just colonize, like generally. They had to stick to one another or adherence. And they had to communicate with one another. So they had to develop mechanisms or proteins for communication or signaling. And so what you see right here is the gene sequence for a protein called cadherin. It is a protein that is involved in adherence. And so you can see that it's found, that is not happening, it's found in choanoflagellates, but there's a new domain that evolved that's found only in animals, but not in choanoflagellates. So perhaps this domain might have been important in that jump from being a single-celled organism, flagellates, to a multicellular organism that attaches. All right, so we are going to learn about a number of different animal phyla. Our common ancestors here, our early diverging organisms include the sponges, the cnidarians, the C is silent, and other organisms called the ctenophores. And so we have a group of phyla that we call the Ediacaran-biota. The Ediacaran-biota, they were early diverging. They evolved in the Ediacaran period. During the late Proterozoic era. And so they evolved before the Cambrian explosion, which happened during the Paleozoic era. So some of these Ediacaran-biota include sponges, cnidarian, and some members of the phylum Mollusca, but many of them have been wiped out from the mass extinction events that have occurred since. All right, so after the Proterozoic era, so here in the Proterozoic era, this is when life, or it's not life, this is when animal life first evolved, and those first animals we refer to as the Ediacaran-biota. in the Paleozoic era, we had a huge expansion in animal diversity. So this expansion was marked by the appearance of several phyla, including the echinoderms, the annelids, which are earthworms and leeches, and the arthropods, which include insects and crustaceans, and the chordates, which include us and other vertebrates. So the first important event of the Paleozoic era would have been this Cambrian explosion, which is about 540 million years ago. You're not going to have to memorize these dates. And then we had land colonization first by the invertebrates about 460 million years ago, and then a mere 100 million years later, vertebrates caught up and made their way to land as well. All right. As we transition to this new topic right here, now we're at the bottom of page 88, and we are starting body plans. We're going to go over a lot of information of the different body plans of different animal phyla. And the reason we're going to go over a lot of this background information is that you will need to know the body plan characteristics for every animal phyla we discuss. So the terms that we learn here are going to be very important in the upcoming lectures. All right. So the body plan consists of 4 different traits: symmetry, the number of tissue or germ layers, the type of body cavity an animal has, and how it develops. And so traditionally, animals were classified into different groups based on these 4 characteristics. But now that we are able to sequence DNA or the genomes of different animals, the groupings have changed a little bit, but the body plan is still relatively consistent in a shared characteristic between many species of the same clades or groups of organisms. So we're still going to learn about those. So we're going to start with symmetry. Animals can be one of 3 types of symmetry. And so our sponges, for example, are asymmetrical. And since we're going to be talking about these for every single animal phylum in the future, I've developed some codes to talk about these or to take short notes. Asymmetrical animals we'll refer to as A. So sponges are asymmetrical, which means no side appears to be similar to the side opposite of it. So if they lack symmetry, they are called asymmetrical. And only our sponges are going to be asymmetrical. Radial symmetry is when every side is identical to the side opposite of it. So mirror images radiate from that center point or the middle. And our cnidarians and ctenophores, an example of a cnidarian is like a jellyfish, are going to have radial symmetry. And jellyfish technically aren't fish, so really we're supposed to just call them jellies. All right, so we'll give this the code R. And then animals that have bilateral symmetry have 2 sides, so a left and a right side, a top and a bottom side. And usually the left and the right sides are mirror images of one another. So they're split between a left and a right or a top and a bottom, and oftentimes both. So there are some terms to go along with bilateral symmetry. In animals that have bilateral symmetry, the top end is going to be the dorsal end. The ventral end is the belly side or the bottom side. The posterior is the back of the animal. And the front of the animal is the anterior side. So those are some terms on your study guide. And also cephalization. When animals become more developed, they have more sensory or nervous system equipment towards the front end or the anterior head of their body, which is called the head. So animals that have prominent heads with a lot of sensory equipment, this happened because of the process called cephalization. Whenever you see that prefix cephalo, it usually refers to head or brain. All right. So the development of sensory equipment towards the head or the anterior region of the animal. So these words or these definitions are at the top of page 89. So continuing on page 89, next we've got the number of germ layers. Sponges, or the phylum Porifera, don't have true tissues. They do have specialized cells, but they are technically not separated by membranous layers, which is what true tissues are. So it's a technicality. So we don't have a code for this. We're just going to say no tissues. If an animal has tissues that develop only from those endoderm and ectoderm layers, we're going to call them diploblasts. So the code I give diploblasts is a D. Whereas triploblasts have a third germ layer called a mesoderm. And mesoderm is going to be that tissue that forms within that blastocele cavity. And this is going to develop into organs, internal organs. The tissues that make up our internal organs, like the heart, the lungs, and the kidneys. So not all animals have hearts, lungs, and kidneys. Animals that do have those types of organs are going to be called triploblasts. And one fun fact to make it easier for you to remember what animals are triploblasts is all animals with bilateral symmetry are also triploblasts. Maybe that wasn't that fun of a fact. All right. Third on our list, down at the bottom of page 89, animals are going to be classified by their type of body cavity. Now, in order to have a body cavity, you have to have mesoderm tissue. So for body cavity classifications, this is only going to be applicable only to our bilateral triploblasts. All right, so we've got three types. We have our acoelomates, which don't have a body cavity, our pseudocoelomates, which sorta have a body cavity. Pseudo means sorta, a means nope. And then our coelomates are our ones that have the true body cavity. So let's start there. A coelomate has something called a coelom. So we have that prefix "cele" again. So this is yet another type of body cavity. And a coelom is a cavity within the mesoderm layer. So in order to have a coelom, you have to be a triploblast because only triploblasts have that mesoderm tissue layer. So the reason it's applied only to triploblasts is because only triploblasts have mesoderm. Which is colored in red here. All right. And so a coelom is a body cavity or fluid-filled space that is fully lined by mesoderm tissue. So a true coelom is fully lined by mesoderm tissue. Our pseudocoelomate has a pseudocoelom, and you can see here it's got a cavity, but it's only lined by mesoderm tissue on one side. The other side is just the digestive tract. So our pseudocoelomates have half a coelom, or they have a body cavity, but it's only lined on one side by mesoderm tissue. Our acoelomates don't have a coelom. You can see here they do have mesoderm tissue. They just don't have a cavity within it. All right, so our acoelomates are going to be a little bit flatter than pseudocoelomates and coelomates, but they do have some internal organs like nervous tissue. They do have mesoderm tissue. All right, this picture is from your lab manual that just shows a two-dimensional cross-section of coelomates versus acoelomates versus pseudocoelomates. And again, you see in the acoelomates, they have mesoderm tissue here in the center, but sorry about that. It's there's no cavity within that mesoderm tissue. And a pseudocoelomate has a body cavity, but it's only partially lined by mesoderm tissue. And here in our coelomate, we have body cavities fully lined by mesoderm tissue. So all of our bilateral triploblasts will be one of these three types according to what type of body cavity they have. Our bilateral triploblasts will also be either protostomes or deuterostomes. So now we are on page 90, and this is going to refer to development type. So we have only two types. We've got protostome development and we have deuterostome development. And like body cavity, this is only going to be applicable to our bilateral triploblasts. One of the reasons is because the first difference between a protostome and a deuterostome is how that mesoderm tissue forms. And so only triploblasts, remember, have mesoderm tissue. Diploblasts do not have mesoderm. They only have the ectoderm and the endoderm tissue layers. And to be honest, I don't care if you know how the coelom forms from the mesoderm tissue, but if you look at the picture, what I can say is protostomes and deuterostomes undergo different types of coelom development, or their mesoderm tissue forms in different ways. Right, so here in a protostome, you see the mesoderm kind of forming within the blastocele separately from the ectoderm and the endoderm. Sorry, the ectoderm and the endoderm. And then here in a deuterostome, you see the mesoderm tissue sort of pinching off the endoderm germ layer and becoming its own specialized layer, and it'll eventually pinch off and form two different body cavities on each side. Humans are deuterostomes. So what I do want you to know about the differences between protostome and deuterostome, you'll notice in your course pack that protostomes are called mouth first and deuterostomes are called mouth second. So what that means is that in a protostome, if the mouth forms first, so that blastopore, which was the entrance to the gastrula, what that means is the blastopore becomes the mouth. And then the anus forms second. In a deuterostome, the blastopore becomes the anus, and then the mouth forms second. So the fate of the blastopore is different in a protostome versus a deuterostome. In a protostome, the blastopore becomes the mouth. In a deuterostome, the blastopore becomes the anus, and then the mouth forms second. All right, we're ready to wrap this up with some summary of the different animal clades, and we're going to relate it to some of the traits that we've just learned of different animals. And so metazoa is the name of the clade. So a clade is just a group of phylogenetically related organisms. So metazoa includes all the animals. Eumetazoa includes everything right here. And they're all related by genetic sequences or by their DNA. So our clade metazoa just refers to all animals. Parazoa, it is not on the list on page 90, but this refers to the animals that don't have true tissues. Eumetazoa, on the other hand, includes all of the other animals, and so these are all of the animals that have true tissues. Then we have our clade Bilateria, which includes everything from the Acela down to the arthropods. And as the name suggests, every member of the clade Bilateria has bilateral symmetry. And they also are triploblasts. Okay. And then within the Bilateria, we've got the Deuterostomia, and they have deuterostome development. We've got the clade Lophotrochozoa and Ectozozoa. So as we go through our invertebrate lecture, it's going to be a really long lecture, and I'm going to split it up into 4 parts. The first part is going to be the early diverging animals. So we call these ones the early divergers. They don't belong to any clade with one another, other than some of them are eumetazoans, and the porifera, of course, are parazoans. After that, we're going to talk about the 3 different clades within the clade Bilateria. And so the second group of organisms we're going to talk about will be the Lophotrochozoa, and these pictures give you examples. So within the Lophotrochozoa, we've got the mollusks, which are snails and cephalopods like squid and octopi. Annelids include earthworms. Platyhelminthes includes flatworms, which is this cute little planaria guy, and also tapeworm. And next, after the Lophotrochozoans, we're going to talk about the ectozozoans. Nematodes and arthropods both have a thick cuticle that undergoes shedding, and they are genetically related to one another. And then finally, our 4th clade that we'll discuss in our last clade will be the deuterostomes, which includes starfish, which are one of our sister phyla. And so the chordates will include some invertebrates, but also our vertebrates as well. So this is where our humans fit in. Us. All right, so before you move on to the invertebrates lecture, make sure you do your Mastering Biology homework. I'm going to put up a quiz for you on Blackboard, and also in your course pack, don't forget, we've got a lot of terms that we just learned, and make sure you review the concepts before you take the quiz. Have a separate sheet of paper devoted to addressing the concepts, and this will be useful for you to use when you're studying for the test and also taking the test. All right, everyone. Hope you have a really good weekend. Bye.